Hello everyone, welcome back with another video lecture on a parasitic disease of horse and donkey. You know, horses are infected with many parasitic diseases. One of the important helminths of horse and donkey is Habronema, which causes habronemiasis or cutaneous habronemiasis. So in this lecture, I'm going to talk about the important species that causes habronemiasis or cutaneous habronemiasis, morphological features, and life cycle of those helminths. Later on, I will discuss the pathogenesis, clinical manifestation, diagnosis, and treatment of habronemiasis. If you like this video, put your comments in the comment section below. Your feedback will be highly appreciated. If you want to get more video lecture on the parasitic diseases of animals, don't forget to subscribe the channel. So let's get started. There are two important genera under the superfamily Spiroideae. They are Habronema and Drashia. Under this genera, the important species are Habronema muscae, Habronema microstoma, and Drashia megastoma or Habronema megastoma. The final host for this parasite is horse and donkey. Different muscid flies such as Musca domestica, Musca autumnalis act as intermediate hosts. The location of these parasites are in the stomach of final host. This parasite is responsible for causing gastric habronemiasis or cutaneous habronemiasis or Sama sore. They may even cause ocular form of habronemiasis. Morphological features of Habronema species. Grossly, they are whitish, slender worms around 1 to 2.5 cm in length, and male parasite have a spirally twisted tail. Microscopic features include two lateral lips, each being trilobed, and buccal cavity is cylindrical in shape. Male parasite has a caudal alley and 4 pairs of precloacal papillae. Left spicule is slender and longer in comparison to the right spicule. Bulba of the female parasite located in the middle of the body. Life cycle of Habronema species. Life cycle is indirect. That is, for the completion of the life cycle, there is involvement of the intermediate host. There are different species of muscid fly such as Musca domestica, Musca autumnalis, Stomoxys calcitans, etc. Final host of this parasite is horse and donkey, and the predilection site of this parasite is stomach of the final host. Infective stage is L3, which will be developed in intermediate host, that is in flies. Adult female parasite is ovoviviparous. They will lay larva containing egg or L1, which will be passed through the feces. Fly will ingest this L1 from the feces during taking meal on it. Later on, L3 will be developed in flies. Then this fly will release L3 to the skin or around the eyes of the host during visiting the host for further meals. Sometimes the entire fly containing L3 can also be ingested by the final host. Later on, the development of the adult parasite will occur in the glandular part of the stomach of the final host. It is also noted that when the larva of the Habronema species deposited on the skin or owned area of the skin or around the eyes, they will invade the respective tissues but won't complete their life cycle. Pathogenesis of Habronemiasis. For the better understanding of the pathogenesis of Habronemiasis, you need to understand the life cycle first that I have already discussed. In this slide, different forms of habronemiasis such as gastric habronemiasis, cutaneous habronemiasis, and ocular habronemiasis will be discussed. In the life cycle of this parasite, you have seen that the infective stage L3 are released on the skin, around the area of the skin. If these L3 stages are swallowed by the host or accidentally ingest the entire fly containing L3, this L3 will reach to the stomach and become adult parasite over the time. This adult parasite will provoke ulcerative catarrhal gastritis. Drashia species have the ability to penetrate the stomach wall to the submucosa and produce large tumor-like fibrous nodule. This fibrous nodule cause mechanical interference of the function of the stomach as they protruded or pedunculated to the lumen of the stomach. The entire thing is known as gastric form of habronemiasis or gastric habronemiasis. When the fly deposited L3 on the moist area of the body, such as skin, any wound area of the skin, around the eyes, they penetrate the affected skin and produce granular dermatitis, which is a non-healing granuloma around 8 cm in diameter. This cutaneous granular lesion further aggravated by self-inflicted damages followed by secondary bacterial infection. So altogether, this condition is known as cutaneous habronemiasis or jack sore or just simply granular dermatitis. This is also known as summer sore, as the incidence of this type of dermatitis is more prevalent in summer season when the flies activities are more. You can see the lesion on the hoof, prepuce, and the urethra, as well as ventral abdomen of the horse. And ulcerative granular conjunctivitis is resulted if L3 stage is deposited around the eyes or on the skin around the eyes. Nodular thickening of the eyelids also be observed. This form of habronemiasis is called ocular habronemiasis. Occasionally, larval stages may also be found in the lungs, and they are associated with nodular peribronchitis. Clinical manifestation of habronemiasis. There are 3 forms of habronemiasis. They are gastric habronemiasis, cutaneous habronemiasis, and ocular habronemiasis that we have already discussed in pathogenesis section. In gastric habronemiasis, there is no specific clinical sign. Heavy infection with Habronema majus or Habronema muscae may cause ulcerative or catarrhal gastritis. Infection with Drashia megastoma may produce fibrous nodule in the stomach, which are pedunculated or protruded to the lumen of the stomach, and these lesions may cause mechanical interference with the function of the stomach. In cutaneous habronemiasis, granular dermatitis is seen in different parts of the body. The lesions are most commonly found on the limbs, prepuce, external genitalia, and ventral abdomen. Lesions are often reddish brown in color, and ulcerative lesion can also be observed. Pruritus can also be seen when horse scratch or chew the lesion. In ocular habronemiasis, conjunctivitis with nodular thickening of the eyelids is observed. For the diagnosis of habronemiasis, clinical findings are important that have already been discussed, and cytologic aspirates or biopsy material from the cutaneous lesion may reveal larvae as well as eosinophil admixtured with different kind of inflammatory cells. An examination of the feces may reveal larvae of Habronema species, which may help in the presumptive diagnosis of gastric habronemiasis. Treatment of habronemiasis. Ivermectin or moxidectin can be given to kill the adult worms in the stomach. In confirmed cases of cutaneous habronemiasis, organophosphorus compound can be given topically to the affected skin. This will kill the larvae. Along with this, fly repellent can also be used to reduce the fly attack to the affected area. Next, based on the lesions and contamination, topical or systemic antibiotic can also be administered. Sometimes topical or systemic corticosteroids can also be given to enhance the healing of the lesions. And finally, surgical removal or cauterization of the excessive granulation tissue may be necessary. These are the references that I have used to prepare this presentation. If you like this video, please subscribe the channel to get more video lecture on parasite of animals. Thank you for watching this video and good luck for your study.